Good evening, everyone. My name is Gina Makwabe. I think I'm audible enough. I'm the consultant physician and nephrologist working in Tanzania with Africa Healthcare Network. I'd like to welcome you all to today's AHN Nephrology webinar. And today's topic is straight from the shoulder, the COVID vaccines. And it's a great honor to introduce our our speaker, who is Dr. Sudhir Krishna. Dr. Sudhir is a professor of biology and biochemistry from the National Center for Biology Science and distinguished visiting professor of biology and biochemistry at IT Goa in Tata Institute of Fundamental Research. Dr. Sudhir is a past chair of Cancer Task Force, Department of Biotechnology at the Government of India. Professor Sudhir is also has a research area of interest is vaccine development, cancer biology, biology medicine interface, and he's a fellowship from Harvard Molecular Genetics Laboratory and PhD candidate from from Cambridge University, UK. He's also a fellow of the Cambridge Commonwealth Society. Professor Sudhir, you are warmly welcome for today's session. We are lucky to have you, and we are ready to hear from you so that you can share your knowledge with people from this area of the world, and we'll be listening from you. So before I allow Dr. Sudhir, I would like to make a few announcements that for those participants from Tanzania, this session will be accredited by Tanganyika Medical Council. So make sure that you send your name and your 3 names and your CPD, Tanganyika Medical Council registration number, and therefore so that you can get this CPD points. So it's only by sending your email, that's when you'll be getting your CPD points. Without doing that, then it won't be possible. So Dr. Sudhir, you are welcome, and the floor is yours. Thank you.
Well, thank you very much. It's both a pleasure and an honor to be with this group. What I want to do this evening is 2 things really. Lloyd, it's a talk about vaccines and COVID vaccines and so on. And I will do that, and I will try and give you a sense of virology and immunology and vaccine perspective so that you have a better sense from a physician perspective. And the other is I want to encourage you to think a little bit about systems and knowledge systems, and how do knowledge systems work, and how do we make them stronger? Like, how do we build vaccine ecosystems across India and Africa and so on? How do you work with silos, and how do you work across silos at the same time? These are some of our big challenges. Silos in terms of discipline, silos in terms of the private-public partnerships, silos in terms of India and Africa, silos in terms of people who work in community and in the lab at the same time. And you need all of them to make vaccines work and so on. And anybody who's even thought about COVID even remotely for the last 2 years would have the same sense of that, that you need all these working as networks from before to actually have it work. And I will hopefully also introduce some of my Indian colleagues, Vijay and Deepti and others who have come on to give you a sense of how I think we can work together going forward. Before I sort of...
Sudhir, you can introduce them too, please. Sorry,
Okay.
It's
so
good to introduce.
I have, I just quickly saw 2 of my colleagues who had come on, Vijay Chandru, and I think Vijay actually came with me on one of my trips to the Kavi Institute a few years ago before COVID. And I'm very keen that you engage with him. Maybe if you can get him to do a talk for a community, that would be very useful. Largely because Vijay has, spanned both the industry and the private space, all the way from engineering, artificial intelligence, computational biology to wet labs. And I think is the kind of person that you would want to help you with the ecosystem. Deepti is actually a younger who's a Drosophila geneticist, but then gives you the other example of what you need, which is basic science. And she wants to do a workshop in Africa using latest CRISPR techniques and so on. So part of my talk is about the fact that we need all these people engaged and talking to each other in different ways. And so hopefully we'll get them on later in the discussion.
Sure.
Yeah.
Sure. Thank you.
So I'll move on to the next slide. Sorry, Katie, how does one move to the next slide? That should work. Well, just 2 lines to tell you that, so my core lab actually works on something different, which is not signaling cancer. And we've been looking at a signal transduction pathway for over 30 years and the pro-oncogenic role in cancer. And that's our core area of research. And we've sort of in the last few years been working on vaccines and so on. And I just thought it's only fair to mention that while I get started. Okay. So a little bit of history, and I think we tend to underestimate the importance of humanities and diverse historical sources in histories about vaccines. So I'm sure if I asked this audience about Edward Jenner, everybody would have heard about Edward Jenner. But way before Edward Jenner actually introduced vaccines about 200 plus years ago, Onesimus was the one who actually introduced variolation in America. And Onesimus was actually an African slave in Boston. And the Ottomans had actually introduced variolation in parts of Africa. And Onesimus actually, when he was in Boston, told the Boston community that there is this technique of variolation, which they didn't believe him. But then, fortunately, the person he was attached to took him seriously. And the first clinical trial in some ways, in fact, if you read some of the reviews, the first global clinical trial of smallpox in some ways was actually thanks to Onesimus, who in one of the Boston pandemics introduced variolation. And of the 242 people inoculated, only 6 died, as opposed to 1 in 7 deaths among the population of Boston who didn't undergo the procedure. And variolation was so important that well before Edward Jenner introduced vaccination to the world, and I think I have a slide after this, yes. In the sort of American War of Independence, George Washington actually got everybody variolated. So it says vaccinated against smallpox, but sort of legally, if you want to be absolutely correct in the use of the term, it's actually variolation. So I actually wanted to show this slide. The previous slide was about Lady Mary Montagu, who introduced variolation in the UK, again from the heart of the Ottoman Empire. I think she was the wife of a major British official and went back and introduced variolation in the UK. The reason I was so keen to show these slides, because there's the heart of one of the most important vaccine stories that we've heard about. And knowledge flows across diverse systems. And we must make an effort over time and effort in different ways to make sure that we have diverse sources of history, diverse ways of thinking about things and so on. And part of, for us, our engagement with Africa, and I'm talking about my team and in India, is to actually learn a lot more from different systems and different knowledge pools, and not just one kind of hierarchical knowledge. So there's so much for the ideological position of where we're coming from, but I think it's pretty reasonable to have said that. And that story clearly makes that point. Now, when you go to vaccines and vaccine histories, actually what's interesting is that you have a convergence of 3 or 4 different themes. And I'm sure in clinical medicine, which a lot of you are engaged with, you see that all the time, which is that you can't wait for basic science. You often have to solve a problem with the tools that you have available now. So if you have a major smallpox epidemic, you have a polio epidemic, you have to create vaccines. And at the same time, immunology and basic science is evolving, and the two of them sort of work together, often in tension with each other. And then at some points, a long history, and I will actually show you how later on in the talk in the context of adjuvants, for example, adjuvants haven't changed over 70, 100 years, and then suddenly something changes. So nucleic acid vaccines, which I'll talk about as we go along, an inflection point has happened during the COVID pandemic, when suddenly nucleic acid vaccines have become very important. So history again has this strange insight or peculiarity that you don't have changes for a very long period of time, and suddenly some event or crisis drives change. And the reason I'm repeating that, or I'm trying to emphasize that, is because I think we are at that crisis moment. And this is an opportunity for us as collaborators, because we, as an Indo-African group, we have a dengue DNA vaccine that we worked on together. And I think this is a great, and we have an mRNA vaccine too. And this is a great opportunity, I think, for us to work together, leverage new technologies, and bypass a lot of sort of roadblocks which have existed in the past. If I can use a crude analogy, in the Indian context, it was often said that we were able to expand landlines only to a certain point, but we were able to scale mobiles, and somebody like Vijay could probably give you the numbers from the top of his head, on a much greater scale because of the way mobile technologies could be used, and and the cost and the private sector and so on and so forth. And so there are moments, I think, in history when there is an opportunity with a new technology, a crisis. I mean, even the fact that we're able to do the Zoom conversation is a new technology. And I think that that moment is now, and we should be working together and getting on with it. So what's interesting about, and this is these pictures that I'm showing of Emil von Behring and Kitasato are actually to make 2 points. And the points are these, and then Karl Landsteiner too, is that in addition to classical biochemistry, and then much later on molecular biology, recombinant DNA technology, immunology has always been driven by people in the medical field who've looked at curious medical observations. So the very fact that, for example, you could not do, the idea of blood transfusion actually came out of the idea that there were these different kinds of blood groups which you don't really know what they were all about. But in animal systems and others, people knew that you could transfuse certain kinds of bloods, but you couldn't, do others and so on. And so this concept of anti-A and anti-B, which becomes the basis of blood transfusion and so on, stems from a clinical observation. And the history of vaccinology, the history of immunology, as I said, has been driven by crisis. You need a vaccine, has been driven by medical observations, clinical observations, and the third pool has been, basic biochemistry, molecular biology, and so on and so forth. And all these are important and must work in an ecosystem with some tension and some competition, of course, with each other. But you need all of them to actually build good vaccine ecosystems. And I was sort of keen on getting that point across, that you can't just do one thing alone, but you must do all of them. This is just to make the point about Landsteiner and so on. So let me give you a quick snapshot of what do we know about immunology. So one of the fundamental, so this is Tonegawa's, so Tonegawa in 1975 got the Nobel Prize for the fundamental concept in immunology, and that is the idea of clonal selection. And one of the hallmarks of immune recognition, and this operates, and I'll first say it in the context of antibodies, and then we can talk about how it's different in the context of T-cell recognition. The hallmark in the context of antibodies is that a single receptor recognizes a single antigen. Okay, so that's your first fundamental rule of immunology. For a very long time, the challenge was, and in this particular lecture, I don't have time to take you through the history of that. The whole question was, how does this happen? And what Tonegawa showed was this amazing sort of observation that in lymphocytes, there's actually a rearrangement where you bring receptors to, where you bring the pieces of DNA which code for the receptors together. So the coding regions are brought, you select for one variable region, bring it across to the constant region, and that's how you generate antibody diversity. And this is a very fundamental point. The reason I'm making this point is because the generation of antigen recognition is independent of the antigen. And so this is something very important in vaccine design. This is something very important to remember in infection and immunity, that you have a pre-existing pool. The initial pool of recognition is completely independent of what it's going to be seen. Of course, there are processes of tolerance and selection processes for both lymphocytes, I mean, both B lymphocytes and T lymphocytes, but this is a very important point to bear in mind. Now, and it's a point that I was telling you about antigen receptor diversity. Now, a couple of points about T cells, because I think they're very important to bear in mind. Broadly, there are three classes of T cells: helper T cells, cytotoxic T cells, and T regulatory cells. What I want you to remember is two things which are very important. T cell recognition essentially occurs at the level of MHC diversity. And so MHC molecules, which, and particularly nephrologists would know this very well because you're always dealing with kidney transplants and so on and so forth. MHCs, major histocompatibility antigens, pick up peptides from inside and put them on the cell surface, and they're recognized by T cells. Okay, so here is your first two differences with antibody recognition. Antibodies recognize whole antigens. T cells recognize bits of the antigens. T cells recognize antigens in the context of presentation by the MHC molecules, both class 1 and class 2. That's a little bit of a detail, and I'll tell you what the detail really matters. But these are the two fundamental points that you need to remember about T cell recognition and antibody recognition. Now, if you want to take a third important point, and this is very important because with the nucleic acid vaccines, they're essentially derived from these principles, is that T cells or peptides are derived from two pools. So if you have class 2, which generate T helper cells, there's just a recycling of antigens which occurs on the cell surface. MHC, the CTLs recognize class 1 peptides, which are derived from inside the cell. Now, why am I stressing on this? Because when I'm going to talk about the vaccines, when we're going to talk about adenoviruses, and when we're going to talk about mRNA vaccines, we can see that mRNA vaccines and adenoviruses can actually generate CTL recognition, which, for example, the inactivated vaccines cannot. But the inactivated vaccines have other advantages, and I'll talk about them as we come along. So that's a sort of nutshell idea, and I'll just to make this point again. Antibodies recognize whole antigens. T cells recognize peptides, which are presented on the cell surface, and these peptides are recognized in the context of the MHC diversity, which exists in all of us. And the MHC diversity is actually critical in some ways to the survival of populations. Because if we were all clonal, we would have all disappeared because of one particular highly virulent virus of other pathogen which would have come along. So that's the sort of nutshell of T and B cells. But I want to leave you with, apart from the idea of networks and memory and so on, which I will come into the context of vaccines, I want to tell you about two things. One of them is that innate immunity, which is another arm of immunity, is a poorly recognized and poorly appreciated immunity. Innate immunity is actually very important. It's because the type of recognition is very diverse, and the kind of recognition is not the classical antigen recognition process where you have clonal selection and recognition. It tends to be underappreciated. The second important thing about innate immunity is innate immunity happens very early. So if you look at the purple graph, which shoots up and comes down by 7 days, that's where you have innate immunity. Now, for a very long time, people thought that innate immunity cannot be very important because you don't have a certain kind of antigen recognition system. There is actually an antigen recognition system for innate immunity. And in a short sort of discussion like this, it's sort of a little hard to cover all that, but I just sort of thought I'd mention it. The other reason people thought that innate immunity is not important is because they thought innate immunity does not have memory. And this is, again, I would like some of you to please think about it and read about it. The process by which innate immunity generates memory is at a molecular level very different from the way clonal recognition occurs. So there's a whole bunch of cells like macrophages and so on, which generates innate immunity. And there is a process of trained innate immunity by which you have modifications in the transcription factors and you have epigenetic control at the level of biochemical modification of transcriptional processes by which you can generate innate immunity memory. Now, these are difficult to work with. These are poorly understood. And so there tends to be a bias for clonal recognition and vaccines and so on. And in fact, I'll come to one of the flaws, not necessarily flaw, but one of the sort of limitations of spike proteins is that they tend to focus on a certain kind of immunity recognition, which the other vaccines don't. So I think I've sort of given you an overview of what the sort of immune status is, and the fact that, the innate cells and the hematopoietic cells, which from the clonal derivative, actually come from the same pool. So that's actually a very interesting but side observation. Now, coming to COVID vaccines, what's interesting is that if you look at the history of vaccinology, there was a remarkable speed in which these COVID vaccines have been developed. So if you look at, for example, when the people started working on other vaccines, or when they first recognized there was a disease, identified the pathogen, I mean, the COVID vaccine story is quite amazing that way. There are broadly 5 kinds of vaccines, and I'll try and take you through each one of them. So there are the spike protein vaccines. The spike protein is the dominant antigen in the coronavirus, the COVID-19, and the mRNA vaccines and the adenovirus vaccines essentially express the spike protein. Okay. The strength of the spike protein vaccines is that you get immunodominance. And as I said, if you want clonal expansion, you want T cell recognition, B cell recognition, all focused on a single protein, the spike protein vaccine is a good vaccine to use. The mRNA vaccines don't have a backbone. So in a sense, you're only generating responses to the spike protein, right? And as I said, the advantage of the mRNA vaccine is that the way you inject them in and they're expressed, they will also be presented to CTL, so you get a complete recognition. So you get a complete recognition, but you get only a recognition to one protein, which is the spike protein. With the adenovirus, you still have that, but with the adenovirus, you have one limitation, is that because you're using an adenovirus to express it and it's got a whole bunch of its own proteins, you will also make an antibody response and a T cell response to the backbone or to the adenovirus vector. So when you do a boost or when you do a secondary immunization or a boost, you'll also get an expansion to the adenovirus. And that's one of the limitations of the adenovirus vector system. And that's why in the UK and other places, in many of the protocols, they started with an adenovirus in some people and then immunized them with the mRNA as a booster to avoid expansion for the backbone, which is the adenovirus vector. Again, these are all spike proteins. So then the challenges that come up is, what about the other proteins? Are there other proteins of the virus which can be used as antigens? And what happens when there are mutations of the spike protein? And that's an issue with the spike protein vaccines. Now, the third vaccine which has been used, and Covaxin, which is one of the Indian vaccines, and I think the Chinese call it Sinovac, are the classical whole-killed virus vaccines. Now, these use vaccines which have multiple proteins. The limitation of, and so you actually get a much broader response. You also get a response to innate immunity, though in the context of Covaxin, which is the Indian vaccine, they actually use a very interesting adjuvant, which was developed by an Indian called Sunil David, who worked in CMC Vellore and is now in the US working on a biotech company. And a classical example of an MD working in, and then he worked at the University of Science. So a classical example of the kind of people I think that we should nurture, people who work across systems and often land up doing very interesting things, like the adjuvants that he developed. Now, a whole-killed vaccine has multiple, so you generate an immune response across the system, T cell recognition, B cell recognition, you will get immune memory, and you will essentially get innate immunity also. The protein subunit vaccines, there are basically two types of protein subunit vaccines. There's one of them which is just currently being licensed in Germany, and that's expressed in a baculovirus system. So you express just the protein. So the people who have vaccine hesitancy about the mRNA vaccines and the adenovirus vaccines are much happier with the protein subunit vaccines, because it's a bit like the classical hepatitis B expression system, where you express subunits of the vaccine. The last kind of vaccine that you can have in this genre of vaccines are peptide vaccines, where you can actually make peptides. And then, of course, the manufacturing and the synthesis of peptides is not simple. And that's one of the reasons why it's taken time for it to take off. So these are broadly the categories of vaccines. Globally, right now, I think in the US and in Europe and other places, the mRNA vaccine has been predominant. In India, it's the adenovirus vaccine developed by AstraZeneca and Oxford. And in, I think, large parts of Africa. We also in India use Covaxin, which is an Indian inactivated vaccine. And in Chinese, I think I've mostly been using the inactivated vaccine with parts of the Middle East. So that's basically about the type of vaccines. Now, what are the issues when you come up with vaccines? Viruses are subject hugely to selection pressures. And these selection pressures are pretty complex, and you can actually, by a lot of sequencing and so on and so forth, pick them up fairly fast. That selections happen, and selections happen in large numbers. And so you have to constantly sequence, and unfortunately, the selection pressures are often in the sites the neutralizing antibodies bind. So you have to constantly double check whether your vaccines are working in the presence of a selection pressure. And you can do structural analysis. This is modeling, but you can also do structural analysis. The paper from South Africa, when Omicron emerged, on showing the kind of mutations. Omicron was, I think, 30 plus mutations, unlike the original the Delta and others, which had much fewer mutations. Now, two or three things are very important in vaccinology. What is the seropositivity prevalence? So one of the slides that I want to show you is the importance of context. And I'm going to come to that in a few minutes. And the reason why I think India and Africa really need to work together, because the way the COVID pandemic has played out right from the beginning has been very different. So there are global issues and there are local issues. And the local issues depend on previous infections. The local issues depend on the kind of transmission patterns. The local issues depend on behavioral patterns. The local issues behave on policy. The local issues depend upon the demographics of young people versus old people. How do you manage them? And there's a range of issues in which, community transmission of viruses operate, vaccine policy operates. And so you can't take generic models from somewhere and just transplant them. Of course, there's global knowledge, but it needs to be balanced with contextual knowledge. And I think that's just as important with a mix of public health, the mix of lab science, mix of policy, and so on. And I hope that will be remembered in this talk. There are indicators that are very important when you do vaccines. One of the most important things that you have to watch in any kind of community transmission is your infectivity versus your fatality. And whether you look at the 1918 epidemic or whether you look at the COVID pandemic right now, this is one of the most important policy determinants that you need to know. What is the outcome in terms of morbidity and management in the context of an infection? It was a little unfortunate that when the vaccines were first promoted, and I don't want to get into any kind of slander out here, that people didn't actually bring up the point that these vaccines that were being pushed actually don't stop transmission. Unfortunately, the assumption was that they stop transmission too. They're pretty poor at stopping transmission. They're very good at preventing morbidity, and they're very good at preventing progression, clinical progression. And so your management decisions are really going to be based on both these two. And I must say that the Africans, but in the context of the Omicron wave, have actually handled it very, very well in terms of spotting very early on that you were looking at a very infective variant in terms of transmission, but you were not looking at a very infective variant in terms of severity of disease. And so the policies and so on were very good. And what's interesting is if you look at the German data, and this brings me to the second point, and that is the point of memory and how do you choose boosters. So if you look at the German data and you look at the COVID vaccine, vaccinated versus unvaccinated hospital admissions, you can see that as the disease progressed, the number of people who were actually vaccinated were falling ill as the time progressed. So what's happening? What's happening is actually pretty straightforward, that even the vaccinated have a memory problem after a period of time. And that's why this whole issue of booster came up. So in any vaccine that you design, one of the questions that you have to bear in mind is how long does this vaccine work? And in what am I going to do when the vaccine does not work? And what's interesting is that a lot of the childhood vaccines that all of us are immunized with actually work over a very, very long period of time. And that's something that, again, we can think about why those vaccines work so well for a very long time, as opposed to, say, the COVID vaccines, which are clearly fairly short in terms of their pandemic. And clearly boosting is one way by which you deal with the pandemic. Now, I just want to take a minute to talk about adjuvants, because I think adjuvants is something, for example, in the mRNA vaccines and the adenovirus vaccines, to the best of my knowledge, they don't actually use an adjuvant. And this is something that we should be thinking about in terms of an area of research. So I talked about these three cells, T cells, CTLs, and T regulatory cells. I would say that if we want to do some frontier science in the context of the Indo-African vaccine program, one area that we should be working on is adjuvants. The other area we should be looking at is T regulatory cells, because I think it would be very interesting to know how vaccines work against T reg cells and how they fluctuate, how they can be managed, and so on. So a combination, I think, is very important as an Indo-African community, that we take some frontline unsolved problems with whatever disease or whatever pathogen that we find interesting, and not only work very downstream in terms of product development and so on, even though that is very important. So I just wanted to reiterate this point that, sometimes in history, change occurs over very long periods of time, and adjuvants is one of those which really, nothing changed for a very, very long period of time. Okay. So one of the areas which I hope you and many of your colleagues will give us insights, because I think we can all learn from you, is what were the reasons that the whole COVID pandemic evolved very differently in Africa. So when you look at the slide of the COVID vaccines at one point, and Africa actually was not immunized on a big way by COVID vaccines. But if you look at COVID fatalities and you look at COVID outcomes, and Nature actually ran an article on this, you can see that Africa did very well in various. When you look at the various waves and you look at the various outcomes, you can see that Africa did very well. And Nature just passed off as saying the power of experience. I think the power of experience, of course, there were previous epidemics in Africa which actually helped build systems, but it also, there are contextual issues in terms of diet, contextual issues in terms of demographics, contextual issues in terms of cross-infections, contextual issues in terms of transmission and so on, which we really need to understand. And this will be very useful global knowledge for all of us. So I hope many of you will spend time putting this together so that we can all learn more about it. Couple of points that I want to make, and I talked a little bit about the importance of history in the context of vaccinology and so on and so forth. It's also very important to have sociologists in the real time right now. I think if you are going to take an approach of building a program in vaccine science and so on, we must have people from the humanities. If you look at something like, vaccine hesitancy, this actually requires people who are trained in understanding communities, people who understand behavior and so on, to be able to dissect and analyze these programs. I would recommend many of you to go into the web and actually download Kate Brigham's romance lecture. So she is the lady who was the chairperson of the task force which developed the vaccine in the UK. This is the adenovirus vaccine principally, but she was also involved in the use of the mRNA vaccine developed in the US for vaccination in this. And she's written so when Lloyd asked me to do this lecture, I sort of did a Google and tried to see what can I learn. And I would encourage you to download this particular link and see what you can learn from that, where she talks about all these issues: skilling, culture, relationship with industry, venture capital mindset, scientific thinking in the bureaucrats, recruitment, dealing with the next pandemic. And all these issues are as important as understanding science. And so the risk of repetition, again, I'd like to make this point that you need to have science and you need to be thinking about policy and you need to be thinking about systems to actually evolve an important contribution in the Indo-vaccine sort of space, as it were. Now, I was not able to figure out, actually puzzled me because I went and looked at the history of vaccination in Africa and versus the history of vaccination in India. And I was a little puzzled as to why the African manufacturing and vaccines has been so poor. And that's something that I'd really like to learn more about. Because in the history of India, we've been sort of manufacturing vaccines now for nearly 100+ years in various forms, in private and public and so on. So that's something that's left me with a puzzle. But I mean, in whatever way that we can contribute towards, I have a slide about the WHO mRNA vaccine efforts and so on at the end. But in any way that we can contribute, either through academia, through biotech, or through various processes, it's something that we would like to partner as a shared learning exercise. Because it puzzles me. I'm actually puzzled as to why the African evolution of vaccine manufacturing has lagged behind the Indian manufacturing. I'm not talking about the West, I'm just talking about the Indian manufacturing. We go back about 100, 150 years in this country through a range of government and other systems. Okay. I want to talk a little bit about our Indo-African program. And our Indo-African program actually evolved from a program that we set up. So I work in a basic science institute, which started in physics and chemistry, in maths, and as part of the nuclear program and so on. And they also have some level of chemistry and biology, sort of evolved much later. And they're not sort of intuitively a base for biomedical research or sort of community or campus research. But we had a grant from the Department of Biotechnology about 10, 12 years ago to set up labs in St. John's Medical College. And this is a classic example of why I think it's worth doing off-campus efforts. So Chitra, one of my younger colleagues, met Gary Dias, who's a microbiologist, who said, look, we have these fevers of unknown origin, and can you do something about it? And Chitra was, sort of getting interested in genomic technologies, and she decided to then sequence fevers of unknown origin, and she set up this whole, so she did one complete dengue metagenomic assembly, and then scaled it and so on, and she had a whole bunch of papers a few years ago with this. So this was a chance encounter based upon the fact that as a basic science institute, we were working in a clinical campus, and we were setting up the process for conversation. And I think that's the same argument that I would make for off-campus efforts and sort of finding the cracks in the institutional spaces by which you can take basic sciences, other efforts and so on, and bring them together and work together. And this was a good example. So this surveillance program, and Chitra then, of course, with Nimans, Vijay, and other people have contributed hugely to the COVID sequencing in this country. We then ran into Narayan Murthy, who's very well known as founder of Infosys, on one of his visits to NCBS. And because we had been doing this dengue sequencing program on the side, Narayan Murthy said he would support a dengue vaccine program. And so we got funded about 3 or 4 years ago. Around that time, it sort of coincided with an ICMR meeting, who they had organized an ICMR meeting in 2016. And the reason why I'm showing all these slides is what I'm saying is that it's very important while you're doing your various things to also have all these institutions and agencies doing their bits with you, and they tend to converge over time into a fairly sort of viable program over time. So, you know, a series of meetings that then did this. The other point that I want to make is that science is pretty multidisciplinary. So this team that we have in Bangalore has Rahul Roy, who comes from the physical sciences and is now in IISc working on dengue. There's Ramkrishna Prasad, who's an infectious disease physician who's also worked with colleagues in Africa quite a bit. Meenakshi and Sadamani, who are computer scientists, who are biophysicists and modelers, who have been working with us together in this program. So I think interdisciplinarity in the context of teams working across silos, basic science to medical laboratories and sort of medical campuses, encouraging more of this is very important. A mix of basic science and applied and so on. And of course, we organized a meeting early in 2018, which we could then take further. So that is when we met our colleagues from the Kavi Institute of Clinical Research, who was set up about 20 odd years ago and have been working in the HIV vaccine space. And we've been working very closely with our colleagues from Kavi. Kavi, of course, many of you would know better than me. I mean, on a day-to-day basis, they have a strong presence in HIV and communicable disease. We ran a number of workshops in Kavi, and that's one way that you get to know each other. So this is crucial members of our team. Arun, who's led the DNA vaccine program, and others when we went to the Kavi Institute, where we did a series of workshops. So we've done two things now with the Kavi, which I think are reasonably substantial. With the Kavi Institute in Nairobi, we've got a surveillance program where we can do, using nanopore sequencing, a fairly in-depth antimicrobial profiling, which can be extended, and the genomics can also be extended to other areas where the genomics is necessary. The second is we have a nucleic acid vaccine program, which we've developed in collaboration, and we have a DNA dengue vaccine, which again, as you can see, is a consortium. And so again, I just want to make the point about the sort of consortium issue. It's challenging, you know, you're working with so many people, they have different kinds of approaches and so on, but I think it's worth it. And I think apart from interdisciplinarity across the silos, and apart from individual science, which remains the cornerstone of basic and other science, you also need to encourage younger people to work across consortiums and so on, because I think there's a huge learning in that process. And so we have a very nice paper, which has actually got a lot of attention. Nature, through their Nature India outlet, actually ran an article on the paper, on this paper. Shweta Raghavan and other colleagues in the lab have also now got an mRNA vaccine. And what we're doing is we're comparing the DNA and mRNA vaccine with traditional vaccines. It's quite exciting actually today, because Shweta and another colleague, a younger colleague in my group, who I'll very briefly refer to her work at the end, which on integrative medicine, she's a chemist, Sanjukta Mukherjee, actually got a Government of India award today, which helps them develop their entrepreneurship potential. And so I think it's again another area which is very important, that we must take some of our younger colleagues, and I know Frank, who spent time with us from Nairobi, went back and now he's actually set up his own company and so on, in part inspired by colleagues that he met in Bangalore who work in the biotech space. So this again makes the point that I talk about, that, you know, we need to also support younger colleagues to set up companies. Colleagues like Vijay Chandra or others can help you mentor them. They can probably set up you need people who can tell you, at least try and avoid some of the mistakes. Some of the mistakes you have to do by your own learning curve. But, you know, there is the possibility of avoiding some mistakes and so on. Now, coming to where do we go from here? I was very happy to see that Kenya is one of the countries, and others for these WHO mRNA vaccine hubs. We, as I said early in the informal dialogue that we were having before this talk, would like to work with various entities to take the next. We got funded yesterday, so we have at least another year's reasonable funding, and we're also talking to a high volume virology network funding, in addition to colleagues in the biotech space to fund us. So I think there are a range of entities that we can work with you to create, to sustain an entire ecosystem. I'll just end with 2 points. One is, I've talked about the fact that you have to break barriers. And one of the barriers that we have to break is this barrier between animals and humans. It's really meaningless. I mean, coronaviruses we know have come from bats and others, so on and so forth. But if you look at all the major epidemics which have happened in the last 40 years, they've all been zoonotic transmissions, every one of them. So I think if we're going further in the kind of systems we are working with, we must break these barriers between veterinary medicine and clinical medicine because it's a very artificial barrier. The last point that I probably would like to make is that we've also been in a project, not with East Africa, but with Cameroon, where a colleague of mine, Sanjukta Mukherjee, who's a chemist, has been working with, sort of plant extracts in cancer and so on and so forth. And I think, again, the integrative medicine space, which is the sort of, building clinical trial capacity, building manufacturing capacity, building supply chain capacity, and so on in the alternative medicine space, and approaching it analytically and in a cool, calm manner, we could probably add a lot. So I hope I've covered a reasonable amount, and I'd be happy to take questions and have a discussion. Lloyd had said 45 minutes, so I think that's where we are. Thank you.
That was excellent presentation, Dr. Sudhir. Would you like to introduce your colleagues, as you mentioned at the beginning of your presentation?
Yeah, I think Vijay is here. He's still there. Vijay, would you like to come on and say a few words?
No, just happy to. Sorry, my camera is not working, but as Sudhir pointed out, I think my experience has been largely on the informatics and genomics side, and very happy to be of any help, particularly the whole sort of pandemic preparedness and genome sequencing aspects. Plus, I think, as we mentioned, been in the biotech entrepreneurship space for about 2 decades, and be happy to mentor as programs develop. Right? I think that's some brief comments.
Yeah, Deepti, if you're around, would you like to introduce yourself?
Yeah, hi everyone, and thank you for the talk, Sudhir. So I am Deepti, and I do genome engineering in Drosophila. I have close to 20 years of experience working with flies, and with Sudhir, we have been talking about how in space of basic research and genome engineering, we can work together in this space. So that's why I'm interested. Thank you. That's all I have to say.
All right. Thank you very much, Vijay and Deepti. Honestly, I'm very much intrigued and surprised by how far Indians have gone towards the development of vaccines. And Sudhir clearly mentioned that Africa has lagged behind in terms of vaccine development. Yes, but now it seems that we are waking up now. You clearly projected a slide where South Africa, Senegal, Kenya, they are now trying to develop vaccines. And in Tanzania, actually, our president announced last week that we'll start vaccine production. So there is at least a light at the end of the channel. Yeah, but Sudha, I have one question. that is regarding mRNA vaccine. And as you said, it targets spike proteins, which comes the issues of like microns, where there is mutations. I don't know how does that work then, because we have mRNA vaccines in the market, Pfizer, that is one of it. But yet, with the development of Omicrons, which is a mutation, we have been told that it can still work very well. So, of course, you get a booster and then it works. So how does that work?
So there have been two arguments. I think there have been two principal arguments for that. One is that nature actually, on almost on the first day when Omicron came up, they ran a modeling exercise to show that T-cell recognition is preserved. And because T-cell recognition occurs at the level of peptides, which are different from the mutagenic region, and T-cell recognition occurs across the genetic diversity profile, T-cell recognition is preserved. The second argument is that you have selection for certain regions, which then immunodominance changes. So when you have an infection with A and B, and when you have an infection with A and C, if you have a diverse pool of antibodies, when C comes along, even if one of the points which were mutated has happened, then you have a change in the immunodominant selection. So in a sense, you can only do modeling and everything up to a point, and you would always have a concern with the spike protein mutations. But there are mechanisms by which you will still get responses because of changes in immunodominance, because of T-cell recognition, and so on and so forth. So you always have to test and see whether your modeling actually works for that.
Thank you. Thank you, Sudhir. It's a time for question and answers. Please, if you have any comment, question, Dr. Sudhir will be ready to respond to our questions or query or comment.
Sudhir, do you think the response in Africa is because of the predominantly an environment where you have an exposure to a lot of zoonotic diseases historically? And again, if you look, there is literature to show that there is a lot of corona already in Africa, other strains of coronaviruses, and also possibly, there are reports of the MERS-CoV antibodies in Kenya as well. Kenya, which has probably come from the Middle East. What do you think? Could all this have contributed significantly to the African response? It is much, it was not really that sort of a pandemic. It has really not hit too many people, and I think the mortality, I don't have exact figures, and we don't have, but it is far less than what we see both in India and outside.
Yeah, Lloyd, all I'm saying is that do good clinical studies. Do studies and publish them, because it's really worth it. Put this information out there. We tend to underpublish from our parts of the world, both India and Africa. And that's, again, something that we need to think about. We need to document a lot more. I think one of the reasons Western science historically has done so well is because of much better systems of documentation, peer review, publishing, and so on than we have in India and Africa. And I think, so encourage you to document what you have. That's the best way of getting it across. And then it's an iterative process, right? When people look at it, challenge it, improves it. So please document these things. Encourage your colleagues to document them.
So, and the other thing is on your India, Africa collaboration with Kavi. Now, are you planning to develop a vaccine there in the Kavi Institute? Is there any sort of thoughts that way?
I don't want to make any off-the-cuff comments now on what we're doing, but we're very serious. And we have funding, and we've talked to additional funders. And we can create a small group of interested people to take this further. But let's not have an off-the-cuff conversation about this, because we're really very serious about this.
Okay, great. And does it take a lot of time to put a whole project in place and then to manufacture?
No, we are ready to roll with a good idea. We can start tomorrow.
Oh, right.
Yeah. That's interesting and exciting. Yeah. So then, you know, as you clearly pointed out, Africa now, most of the countries are interested in vaccine manufacturing. Actually, not only for COVID, but all the vaccines. So the approach, what would you think would be the best approach? For now, we have heard that they are planning to start manufacturing plants, but I don't know how. Would it be first to train people to come with the knowledge and then come back and establish these factories, or bring people who have already has knowledge and start, you know, I don't know, how would you advise, especially the governments who are intending to, yeah.
So I actually went down to Bharat Biotech, which is the company which makes Covaxin, the Indian inactivated vaccine. Because, you know, they've been making vaccine, they have 18,000 people, I think, who work for them and, you know, so on. I would be able to help by bringing in people who manufacture vaccines. I mean, I understand the vaccine science component, I sort of, you know, but I would just bring in people who manufacture vaccines to answer that question for you. Because they know the issues of scale, they know issues of manufacturing, they know issues of policy, which I honestly don't know. But I will help by bringing such people with you. And as I said, I actually met the Bharat Biotech lot about a month ago, precisely for this point, to get them engaged with East Africa on vaccine manufacturing. Goes back to the point that I was making to Lloyd, that we create a group of people who bring the specific expertise and let them take it further.
Yeah.
Yeah, that's true.
Yes.
I'm sure
That's very
really, yeah, I'm sure there are a lot of countries very interested, you know, to sort of work with these groups, for sure.
Yeah. Any other question, comment from the audience? Okay. All right. Maybe the last questions to Sudhir. Among the vaccines that we have, the mRNA vaccines, DNA, I mean the other vaccines, what would you recommend would be the best vaccines? Because you have like JJ and other.
Yeah, I mean that's a tricky question. Well, I decided to get I took Covaxin, the inactivated vaccine, because I thought that it made more sense. But I wouldn't make a strong argument against one or the other. I would just leave it at that.
And when you use, you know, one vaccine from one company, and then you have a booster from the other side, would you get a sort of a varied antibody response which is beneficial, or is there any benefit or no benefit?
So it's important to understand that mix and match in vaccines. Vaccines are not drugs. And just like you can have an infection and then a vaccine, and you can get vaccinated in infections, you can use vaccines across companies. You just have to look at what the construct is and what the construct is doing. But this notion that you cannot mix and match is just completely wrong. With vaccines, it really doesn't matter. You have to look at what the immune outcomes are.
But is there any evidence to show advantages on cross-vaccination using different vaccines?
I actually said that, right? Because the original recommendation in the UK, I think, was with the adenoviruses to use the mRNA vaccines as booster, because they wanted to cut down the background against the backbone. So there's good examples out there.
Okay, thank you.
All right, thank you. Let me see if there is any comment from the chat box. I don't see any question from the chat box. If there's any, yeah. So if no more question, I would like to take this opportunity to thank you, Dr. Sudhir and your team, Vijay and Sudhir, and Deepti and Vijay. Thank you for your excellent presentation, and we have learned a lot from your presentation. And I would like to thank you for taking your time, prepare this presentation, and share with us. Yeah, so we really appreciate for your effort. Thank you very much.
Well, thank you from our end too. Look forward to seeing you all in person one of these days.
All right, and also I'm very much, you know, excited with the opportunities that you have given us. that you are ready to share and, with us and collaborate with us in terms of the issues of vaccine development. That is very exciting, and I think we should take it positively